Hello students, welcome to EPG Pathsala. I'm Dr. Hari Mohan, Assistant Professor, Center for Medical Biotechnology, Maharshi Dhyan University, Rohtak, Haryana. Today we will discuss the module DNA sequencing under paper Animal Cell Biotechnology. The objective of this module is to tell you what is the significance of DNA sequencing, how it evolved during course of time, what are the latest development in the field of DNA sequencing, and what are the ethical issues involved in DNA sequencing. All organisms on this earth, except the non-living things, possess genetic material. It can be DNA or RNA. In most of the cases, it is DNA. Whereas in some viruses, it is RNA. So whether it's DNA or RNA, they have certain kind of clusters of information that we call gene. And if we are able to decode these clusters, these hidden information, then they will be of much use for mankind. So this has led to development of a concept that is known as DNA sequencing. So what exactly, how we can define it? DNA sequencing is to determine the sequence in which the nucleotides are arranged. So why to sequence? What is the need? DNA sequence can be used for identifying the disease causing organisms. They can be used to identify from where they have evolved, and they can be used for developing diagnostics. So we can say the sequencing is important in ascertaining the phylogenetic evolution of microbes, in setting down the disputes of parentage, disputes of criminal cases, and it is also getting more and more important with the recombinant DNA technology. So there is an urgent need to understand the concept of DNA sequencing and develop latest techniques for it. There are many techniques available for DNA sequencing. So with the age of development, these can be classified into three types: first, second, and third generation of DNA sequencing methods. First generation include the first and second method, that is Sanger's Dideoxy chain termination method and Maxam and Gilbert method of DNA sequencing. So these were primitive methods, suffered with certain kind of disadvantage, and that led to development of second generation methods that are pyrosequencing, Solexa method, solid method, DNA nanoball method. So these methods are relatively quicker and less time consuming, less costly as compared to the first generation method. Now people are going for third generation method, which are in active state of development, and these include single molecule sequencing and single molecule real-time sequencing, microscope based sequencing, and MALDI-TOF MS based sequencing. So these three generations of sequencing, they have provided us a valuable tool, that is DNA sequencing. It is very interesting to know how the DNA sequencing evolved. So it was in 1870 when Messrs discovered DNA. In 1940, every demonstrated that yes, it is DNA which is the genetic material. In 1953, Watson and Crick proposed the historical double-stranded helix model for DNA. So it was in 1953 where the Watson and Crick proposed the model, and in 1965 first attempt to sequence the genetic material was taken. It was Holly who sequenced the tRNA of yeast. Specifically, it was alanine tRNA of yeast, which was sequenced in 1965. So it was almost 13 years between the discovery of double-stranded model of DNA and the first attempt of sequencing. It can be attributed to many factors. When we compare this thing with protein, the sequencing of protein was done much earlier than that of DNA, because the structure of DNA is very much similar to each other. So it was very difficult to separate the very identical DNA molecule. In addition, the proteins are made up of 20 amino acids, which are very different physiologically, biochemically, and structurally, whereas DNA or RNA is composed of only 4 nucleotides, that is ATGC, or in case of RNA it is uracil. So that's why there was much delay in sequencing of DNA. So it took almost 15 years to develop strategy for sequencing the genetic material. So at that time, in 1970, it was all manual. It was who sequenced lambda cohesive and DNA. It was in 1975 when Sanger proposed the first model for DNA sequencing, and that was plus minus sequencing method. In 1977, Maxim and Gilbert proposed chemical degradation method of DNA sequencing. In the same year, in 1977 only, Sanger proposed another model of DNA sequencing, that was Dideoxy chain termination method of DNA sequencing. So with the advent of Dideoxy chain termination method, it overtook all other methods because it was easy to perform and it avoided the risk of radioisotopes. Therefore, after its invention in 1977, Sanger dideoxy chain termination method was very popular, and due to this importance and popularity of Sanger's method, Sanger was awarded Nobel Prize in 1980. So Sanger is the only biologist who got 2 Nobel Prize, one for protein sequencing and other for DNA sequencing method. So here we can see the number of base pairs are increasing and how they are increasing. In 1980, it was Messing who cloned the DNA in the M13. So cloning vectors were used for cloning and subsequent sequencing of the target DNA. Here in 1980 only, the computer were used for the analysis and storage of DNA sequence data. So here we can see the number of base pair are increasing. It is 20 into 100 base pair, then 30 into 250 base pair. It was in 1990 where very important event happened. The step towards automation was taken, and it was very important step. Before that, the DNA sequencing was a manual affair. So everything was manual. You have to run the gel and then you have to visualize it. You have to note down the sequence. But towards automation, it was Hood and co-worker who proposed that yes, this process can be automated. So the automated sequencers were invented in 1990, and with the invention of automated sequencer, better sequencing enzymes were developed. The fluorescent dyes were used instead of radioisotopes, so that decreased the health hazard and increased the efficiency of detection, and high throughput tools with robotic operations were used. So all this helped in increasing the size of base pair that could be sequenced. So here we can see in 1990s 182 into 500 base pair could be sequenced. Then came the era of complete genome sequence. It was in 1997 when the yeast Saccharomyces cerevisiae complete genome was determined. So it was a historical event, means complete genome of yeast was given for the first time. Then in 1999, human chromosome 22 was completely sequenced. Then C. elegans was sequenced. In 2000, the complete genome sequence of Drosophila was proposed. Vibrio cholerae, which cause cholera, was sequenced completely. Human chromosome 21, which is associated with Down syndrome, was sequenced completely. So in 2000 only, the framework of human genome was proposed, and very soon in 2003, the complete human genome was sequenced and published. So that was a historical event when the complete human genome sequence was completed, and this opened a new era of personalized medicine and more screening of diseases due to defects in the nucleotide sequence. So why we need to go for DNA sequencing, or what is the need of DNA sequencing? It is significant in forensic sciences, molecular medicine, evolutionary study, and genetic engineering. In forensic science, DNA sequencing have given the mankind an important tool to catch the criminals by using their blood, by using their hairs at the site of crime. So very easily the criminals can be captured by using the samples at the site of crime. Second, very important is to determine the paternity. So there many times there are conflicts about paternity, so that can be solved using the DNA sequence. Identity of person in case of accident. Sometimes due to accident, the person could not be identified. So if the DNA sequence or DNA profile is available with us, the identity of the person died in accident can be ascertained. In molecular medicine, the DNA sequence has been instrumental in determining the disease gene. Now the scientists have come across a very interesting finding that whether it is a pathogenic disease or it is a genetic disease, it is gene which is responsible for the disease. So if we can identify that gene, it would be easy to treat the disease. Second, very important, it is important in gene therapy. So correct gene can be inserted in the patient with some defective gene. It can treat any abnormality in the embryo before birth. So it is important for prenatal diagnosis or prenatal counseling. So if any embryo is suffering with certain kind of major chromosomal defect, so decision can be taken for termination of pregnancy before birth. And last and very important is concept of personal medicine or personalized medicine, with the idea that every human being is different because their DNA is different, and that's why they respond to the medicine differently. So in near future, it may be possible that for every human being, different kind of medicines can be used that will very efficiently treat the disease. DNA sequence can be used for ascertaining the evolutionary pattern from where a pathogen has evolved, or what is its relation with the other organism, whether it is similar to a particular microbe or it is distant to a particular microbe. It can be ascertained with the help of 16S rRNA sequence in case of bacteria. DNA sequence are instrumental in determining the past event. So when human being was not there on this earth, what happened at that time, what happened with the organisms at that time, that can be studied with the help of DNA sequences. And it also helps in determining the evolutionary changes in the individual gene, how genes which are important for life changed during course of evolution. In recombinant DNA technology or genetic engineering, these DNA sequences are important as we can create the desirable change in gene if we know what is its sequence. it can help in creating biopesticide, which is a major problem nowadays, pesticide. So this can help in eliminating the problem of pesticide, and by using the DNA sequence of natural pesticides, we can insert these sequences in the existing crops and we can create genetically engineered crops. For example, BT cotton is a major success of recombinant DNA technology, and this could happen only because we knew the sequence of BT. So DNA sequencing will play vital role in genetic engineering also. Sanger method of DNA sequencing is based on the principle of sequencing by synthesis. Here, dideoxynucleotides are used, and incorporation of these dideoxynucleotides will result in termination of chain. So this is the basic principle on which the dideoxy chain termination method of Sanger is based. With the advent of this method, it became very popular because it avoided the hazard of radioisotopes and autoradiography, which was the main component of Maxim and Gilbert method. So this method ruled the scientific world for almost 30 years. Up to 2005, this was a major method for DNA sequencing before the next generation sequencing was developed. In Sanger's dideoxy chain termination method, there is requirement of single stranded template DNA, primer strand, DNTP, and one DDNTP. Because this DDNTP don't have the 3' OH group, that's why wherever this DDNTP will be inserted, the cycle of elongation will stop there. So the chain will terminate wherever this DDNTP is inserted. In this image, the cycle will stop where this DDGTP is inserted because it don't have the required 3' OH group. So for each well, there is a single DDNTP. For example, in one well, there is DD-ATP. In second well, there is DD-GTP. Third, DD-TTP. And fourth, DD-CTP. So in 4 wells, these dideoxynucleotides are added, and there are chances that they will be inserted at every position in the DNA strand. So wherever they are inserted, that will result in termination of chain, and these DDNTPs, they are labeled with fluorescent dyes. So when these reactions they are run in capillary electrophoresis, they can be read by laser beam, and that will give the required sequence of nucleotides. So this is a very popular method because of its simplicity and accuracy. So when you will go for sequencing of DNA, you will get these peaks of different color. So these peaks need to be analyzed, whether peaks are of good quality or you need to go for the sequencing. So for determining the quality of sequence data, it is very important to analyze the peaks, and quality of data depends on purity and concentration of template DNA, on presence of extra PCR band, and on quality of the dye terminators, on quality of capillary electrophoresis matrix, and the reagents used for sequencing. So the researchers should convince that these base calls are robust, that non-specific bands are not there, and peaks are very clear. Maxim and Gilbert method was developed in the same year as that of Sanger's dideoxy chain termination method in 1977. Maxwell and Gilbert method is based on the chemical degradation of oligonucleotide or chemical degradation of DNA molecule. So it involved labeling of that DNA molecule with the radioisotope and then degrading the nucleotide at specific position by using specific combination of chemicals. So this method was successful initially, but due to relation with the radioisotopes and time consuming, labor intensive, the method was not very popular and it was not used frequently for commercial purposes. Second generation method. So after this Sanger's Dideoxy chain termination method and Maxwell and Gilbert method were given, people want some methods which are quicker, which are more accurate and which are less costly. So that gave rise to concept of second generation methods of DNA sequencing, and these methods, they can be used for longer sequences. They are less time taking, they are more accurate as compared to the first generation methods. The first second generation sequencing method is pyrosequencing. Pyrosequencing is based on the idea of sequencing by synthesis, same as that of Sanger's dideoxy chain termination method. So what happened here, when DNA is synthesized, pyrophosphates are released. These pyrophosphates, they can be used as substrate for ATP synthesis by using an enzyme ATP sulfurylase. This ATP then be used by the luciferase enzyme and will generate light. This light can be captured by camera and that will indicate what kind of nucleotide is inserted at a specific position. So one nucleotide is added at one time. So this is the beauty of this technique. For example, in this case, adenine is added, which is complementary to thymine, and this DATP, when it is inserted in complementation to the thymine, it will release pyrophosphate, and this pyrophosphate is converted into ATP by using enzyme ATP sulfurylase. This ATP is then used by luciferase enzyme, which emit light by using ATP. Here we can see the non-specific nucleotide. For example, DCTP in this case is degraded by apirase. So only the specific DNTP will give signal in this case, and non-specific DNTPs will be degraded. That's why this make the pyrosequencing more accurate and less time taking as compared to the first generation methods. The first second generation sequencing method is pyrosequencing. Pyrosequencing is based on the idea of sequencing by synthesis, same as that of Sanger's dideoxy chain termination method. So what happened here, when DNA is synthesized, pyrophosphates are released. These pyrophosphates, they can be used as substrate for ATP synthesis by using an enzyme ATP sulfurylase. This ATP then be used by the luciferase enzyme and will generate light. This light can be captured by camera and that will indicate what kind of nucleotide is inserted at a specific position. So one nucleotide is added at one time. So this is the beauty of this technique. For example, in this case, adenine is added, which is complementary to thymine, and this DATP, when it is inserted in complementation to the thymine, it will release pyrophosphate, and this pyrophosphate is converted into ATP by using enzyme ATP sulfurylase. This ATP is then used by luciferase enzyme, which emit light by using ATP. Here we can see the non-specific nucleotide. For example, DCTP in this case is degraded by apirase. So only the specific DNTP will give signal in this case, and non-specific DNTPs will be degraded. That's why this makes the pyrosequencing more accurate and less time taking as compared to the first generation methods. Solexa method. Solexa, which is now part of Illumina, was founded by Shankar Balasubramaniam in 1998. And it is based on the principle that DNA probes are attached on the plate with suitable polymerase, primers, and reversible terminated dye bases on the slide. So we can say Solexa method is based on the principle of reversible dye terminators. The fluorescent nucleotides with reversible terminated dye are used. After binding, the image will be captured with the help of camera, and then it will be removed from that site. 4 nucleotides are labeled with different fluorescent colors. Here we can see that on the base plate, the clonal DNA clusters, they are arranged, and here the single nucleotide which bind to the complementary nucleotide will emit the light, and that light is captured with the camera, and that is the basic principle of Solexa method. Solid method was given by Applied Biosystem, which is now part of Life Technology, and it is based on the principle that different oligonucleotide units attached with different signal molecule are added with ligase. So if binding happens between the target sequence and the fluorescent labeled oligonucleotide, then signals are checked, signals are emitted. It can detect which particular base is present on that targeted sequence. So in this way, the complete DNA strand can be sequenced. Here we can see that different oligonucleotides labeled with different fluorescent dyes are allowed to attach to the target DNA, and wherever these oligonucleotides will attach, that ligation site can be identified, and identification of that ligation site forms the principle of solid method of DNA sequencing. Third generation methods are in active state of development. They are faster, cheaper as compared to previous 2 generations. The important third generation methods are single molecule sequencing, single molecule real-time sequencing, DNA nanoball sequencing, and nanopore based DNA sequencing. Single molecule real-time sequencing is an example of third generation sequencing methods, and they are in active stage of development. Here, as the name indicates, sequencing is done in real time. It means sequence is determined when DNA is synthesizing. In this method, zero wave mode, that is ZWM, is used. This zero wave mode is a nanophotonic hole on silica plate coated with aluminum cladding film. Many ZMW are present on silica plate with single polymerase and single DNA strand. Here it is important that the polymerase is immobilized on the bottom of the well, and the signal is read only what happens in the bottom. When light illuminated from bottom, a visualization occurs from where we can observe change or which nucleotide is binding on the strand. So the reaction will occur only when the polymerase will bind a particular nucleotide at the bottom. Fluorescent dye labeled nucleotides are used after formation of phosphodiester bond. dye will be released. So here we can see this method is also based on reversible dye terminators. This dye will give a signal which is detected by a detector. Here we can see these small valves, and on the bottom of valves the polymerase and immobilization of polymerase is key to the single molecule real-time sequencing. Whatever is happening at the bottom can be visualized as sequence of nucleotide. So with the invention of different technology of DNA sequencing, major sequencing facilities have been established, like Craig Venter Facility, Tiger, the Institute for Genomic Research, Wellcome Trust, Sanger Institute, JGI Institute. So these institute, they have massive facilities for sequencing the whole genome. Nowadays, the whole genome sequencing has become cheap. From millions of dollars earlier in 2003, it have come down to only $1,000 till date. So due to these massive facilities for DNA sequencing, the whole genome analysis have become easy because of increasing automation, because of higher throughput and less cost per reaction. DNA sequencing seems to be an interesting technique and very useful also, but there are ethical issues involved in DNA sequencing also. In a famous case of Moore versus Regent of University of California 1990, it was established that the cells of an individual cannot be discarded or cannot be used for profit without the consent. However, with the consent of individual, the cells can be used for profit. In addition, the whole genome sequencing of individuals have made it possible that this data may be stolen and can be misused. The persons can be discriminated on the basis of their genetic information. For example, an insurance company may charge higher premium from an individual with a inferior DNA sequence data, or the person who are more susceptible to disease on the basis of their genetic information may be discriminated on the basis of DNA sequencing. To prevent this, in USA, Genetic Information Non-Discrimination Act has been passed. So people cannot be discriminated on the basis of their genetic information, because the whole genome sequencing is getting much cheaper and every person can afford the whole genome sequence. So it is possible that people may suffer this kind of stress if they come to know that they can get disease easily, or there are chances that they can get cancer in the lifetime. So they may be under undue stress. Although some studies have indicated that it will not put any extra stress, but there are possibility that with the information available to every individual, ethical issues may arise with DNA sequence. Even after third generation of DNA sequencing methods, many new methods are in development, and interesting of them are tunneling current DNA sequencing, which is based on the principle of change in current when DNA of different sizes passes through the nanopores. Second very interesting method is MALDI-TOF MS. Till date, MALDI-TOF MS is used only for sequencing of proteins, but whether it can be used for sequencing nucleotides also is a matter of question. But people are trying to use MALDI-TOF MS for DNA sequencing also. Third and very interesting modification of Sanger's method is microfluidic Sanger sequencing. So it's based on the same principle of Sanger, but it has employed microfluidics. and this use of microfluidics has decreased the cost per reaction and increased the accuracy of sequencing. The fourth interesting mode of sequencing is microscopy based technique. So in this technique, by use of electron microscope, the DNA can be visualized, and by using electron microscope, exactly what is the size and what nucleotide is incorporated could be identified. So these methods are in a state of development, and they may change how DNA sequencing happens today. In the last, to summarize the module, we can say that the first generation method, that is Sanger's dideoxy chain termination method and Maxam and Gilbert method, have paved the way for a very important technology. Later on, Solexa method, uh, nanoball method, Illumina method, and other second generation and third generation methods have improved the efficiency of this technology, decrease the cost, decrease the time, it make it labor intensive. So DNA sequencing has the potential to change the human civilization and the way we are treated. This is a step forward towards personalized medicine, where on the basis of DNA sequence, different medicines can be prescribed to different individuals. DNA sequencing will also help mankind to solve the criminal mysteries, to solve the forensic issues, and to determine how we evolved exactly. It will also help in determining where we are going. So with the advancement in technology, with the decrease in cost, it may be possible that in very near future, every individual will have its DNA sequenced or have its DNA fingerprint, which will be very important for prediction of disease, for prediction of cancer, and making a healthier world. But we have to be cautious that the ethical issues are not overlooked. Otherwise, it can be some kind of disaster to mankind also. But overall, it looks that it is a boon to mankind. Thank you very much.